Okay, so this is going to be a quick overview of genetics and meiosis, excuse me, for to ensure that you understand what the connection is in preparation for your exam this week. So here are two questions that really this podcast is meant to investigate. How are genetics and meiosis related, and how does this connection play a role in my deeper understanding of genetics? So there are a couple of things that we need to talk about, excuse me, to begin with. And one is, what is a gene? And I'm not going to define this for you word for word, because this is something that IB defines for you, especially in your book. So make sure you look at that, and it's one of your targets, your assessment statements, and something you wrote for your notes. You need to memorize that word for word. So a gene is basically a section of DNA. So it's a section of DNA that codes for mRNA, that codes for an amino acid sequence, which is a protein or a trait. And really this DNA comes to you from your parents. And that's really what a large connection of this meiosis and genetics is that meiosis, we're talking about different combinations of genetic material forming into the sex cells or gametes or sperm or egg that are going to recombine to make to come together to make the first cell of the next generation, and in the context of you, the beginning of your life, or what will become your life. So it's quite important to understand that. So if we're talking about a single chromosome, we're talking about two sections, two molecules of DNA that are connected by a centromere. And remember that these two are sister chromatids. And these are made through DNA synthesis during the S phase of interphase with the aid of two very important enzymes, helicase and DNA polymerase, and therefore nucleotide for nucleotide, each molecule, monomer or subunit of DNA is identical unless some kind of mutation occurred, a base mutation or a frame shift mutation. But we need to understand that zooming in on one small section of this chromosome, we would be looking at a sequence of many nucleotides, or DNA nucleotides, which are made, remember, out of deoxyribose, phosphate, and a nitrogenous base. And so if we're looking at many genes, it's often useful for us to talk about a single gene on this chromosome. But remember that this is only one chromosome, and we actually have, as humans, we actually have 46 chromosomes, but they come in 23 pairs, and we call those pairs homologous. And very interestingly, if you think about this, to extend this to all other sexually reproducing organisms, all sexually reproducing organisms have an even number of total chromosomes, or an even diploid number, because that diploid was made from two haploids coming together. And no matter what two numbers, as long as it's the same number, come together, you're going to get an even number. So 11 plus 11 is 22, or 10 plus 10 is 20. So the diploid number is always going to be even, whereas the haploid number could be even or odd. And in humans, it's an odd number. So let's talk about these homologous chromosomes. So let's say that we have a cell that has these chromosomes. And I guess I'll go ahead and draw our cell membrane around it. So let's imagine we've got this cell. There's no nuclear membrane. Nuclear membrane has gone away. And remember, if there's a nucleus, or if we have chromosomes like this that can condense onto this egg shape, we're talking about eukaryotic organisms. And this DNA is wound around histone proteins to make nucleosomes, and it continues to wrap around until it makes these thick fibers, kind of like yarn, that are visible to us under a microscope. But we're talking here about an organism that's got sister chromatids that are outlined in white here, and then let's use another color, let's use purple. These chromosomes, this one right here and this one right here, are homologous chromosomes, and that is because I denoted these in color. Blue came from Dad, red came from Mom, but they contain genes that are at the same locus or the same position, but they may have separate alleles or versions of the gene, which means that they have a different nitrogen-based sequence, which means that if they have different DNA nucleotides in the sequence, they'll code for different mRNA nucleotides and therefore carry a different message, possibly. Remember the wobble, look up the wobble effect if you don't remember it, but it'll carry a slightly different codon message to the ribosome, which will read the codons differently, pull in different anticodons, and therefore different amino acids, and the amino acid sequence makes for a certain protein or trait. So if we change the DNA nucleotide sequence, we're changing the protein sequence, or the amino acid sequence, and therefore the trait. So let's say Dad is going to give one trait, and Mom is going to give, so one allele, and Mom is going to give another. For the sake of demonstrating differences, we're going to use a capital A and a lowercase a. So when we're talking about these alleles, sometimes we refer to them as dominant and recessive. And if we think about what's going to happen in meiosis, in meiosis 1, this tetrad is going to separate. So we're going to end up with two separate cells at the end of meiosis 1. We're going to end up with two cells. Let's see if we can draw them here. And I'm going to add this in just a second. Let's remember that because these sister chromatids are identical, we've got a big A over there and a little a over here. So these two cells, oops, made a mistake there. Erase that. Got a big A, and then we've got little a, little a. That's the end of meiosis 1. But remember that sister chromatids are going to separate after, at the end of meiosis 2, and therefore we're going to have 4 cells that are each going to have one of these sister chromatids. This is going to be big A, little a, little a. And notice that these have separated, and so now we have two cells with big A, and two cells with a big A each, and two cells with little a each. And this is really the simplest model here. But let's imagine that we've got another trait, another allele that we're following. Actually, I'll come back to this in just a second. So if we think about what's happening here, we know that this individual that started this meiosis is heterozygous, because the genotype that this individual has is big A, little a. Now we could actually write this in a way that's a little redundant, like this, but it's going to be equal odds on our Punnett square. So we go ahead and scratch them out, and let's say that I'm going to open up a new slide here. Let's take that heterozygous individual and cross that person with another heterozygous individual. What we're doing here is actually taking one of the, let's say this individual is the male and this is the female. This individual is producing half sperm with this big A allele and the other half with a little a allele. This female is producing an egg with a big A, half of them with a big A, half of them with a little a. And so what we're doing here in each square is putting in the result of fertilization. If this sperm and this egg came together, we'd have an individual for this single gene that would be homozygous, or heterozygous, or homozygous recessive. And so that's really what a Punnett square represents. But this is really a mono-hybrid cross. Mono meaning one. Okay? And really when we're looking back at this diagram, it's a very simple model. We only have one homologous pair. Okay? And we know that we don't have this. So really when we're talking about something a little more complicated, we're talking about something that's got different size chromosomes in it. And when we talk about a single gene, we're talking about one gene on one chromosome. And what's interesting is that when we think about what's going to happen in meiosis, let's go back to this diagram. What happens in meiosis, if we have more genes, like let's say, sorry about that, if we have another gene here that we're following, they're actually going to be traveling together on these sister chromatids. And what actually is even more interesting is that if crossing over happens, we've actually mixed those up a little bit. So we add more variety. And what's interesting is if we go back to this diagram, I've drawn all the blue chromosomes on the left and all the red ones on the right, indicating that all of Dad's chromosomes are on the left and all of Mom's are on the right. And that's really not how it works. Is that really what happens is when these homologous chromosomes line up on the metaphase plate in meiosis 1, when the tetrads form, they form randomly, as far as we know. So the fact that Dad's chromosomes on the left and Mom's on the right is almost like the result of flipping a coin. Which one is going to go on the left and which one's going to go on the right for each pair of homologous chromosomes. So this one could be this way, this could be flipped the other way with red on the left and blue on the right, this could be vice versa, and each time it presents itself the new ratio. It's like flipping a coin. So because there's an option of left or right, we use a base of 2 and we raise it to however many homologous pairs there are. So because humans, again, have 23 pairs, homologous pairs, we say that actually that the probability of any genetic combination is actually, it's greater than 8 billion. So the chance of the genetic combination of all those alleles is quite complicated, and the chance of those genetic alleles coming together in the sperm or the egg, or the egg that then came together to make you, is quite rare, which is fantastic. It makes you very, very special. But it also means that had another sperm or another egg been fertilized, you could be different, which is kind of neat to think about as well. But if we're following different alleles on these chromosomes, it becomes more complicated. And if you think about dihybrid crosses, let's open up a new. So dihybrid, if you remember these from 9th grade, oops, let me erase that. These are the ones that have 16 squares. I don't know if you did these in 9th grade or not. Let me just show you this so we can have a quick discussion, or I can at least point out a couple of things. Alright, so here is my 16 square Punnett square. And now if we're talking about two separate genes, for an individual, we're actually talking about 4 letters. 2 genes, and each has 2 alleles for the genotype. So let's talk about T's and A's. And so this individual is heterozygous for both genes. And remember, we have thousands of genes, so we're only talking about 2 in this situation. So this would be a dihybrid cross. And let's say this person is crossed with another individual that's also heterozygous for both alleles, for both genes. There's an assumption that's made in these kinds of crosses, and that is that these genes are on separate chromosomes, and that's not always the case. Many times the genes are on the same chromosome, so it doesn't get all the combinations. But what we do is basically FOIL, where we do, if we imagine, we'll do this in green, imagine parentheses, we FOIL is what we do. So the first letters, the outer letters, the inner letters, and the last letters are combined. So we've got big T, big A, big T, little a, little t, big A, little t, little a, are the different combinations of these alleles that are going to be produced in the sperm or egg. And because the other parent is the same combination, it's going to have the same outcomes from FOIL. And then all we do is combine the letters as if they were fertilized to know what the probability is for each combination. And the big thing here is really understanding that this is based on this assumption that these genes are on separate allele or separate chromosomes. And I'll let you can fill these out if you want, but that's assuming that we've got A's over here, and let's say, sorry about that, and we've got T's over here. But sometimes those T's actually are here, or whatever letter we want to use, and so they'd always stay together unless there was crossing over. And I hope that that kind of makes some connections for you between genetics and meiosis. And remember that this drawing has a diploid number of 6. That's the diploid number. But really for humans, what we would see is 23 pairs lined up. But we don't ever draw that because it's just too complicated. And know that we would actually see 23 pairs lined up, 22 of which are called autosomal, and the last pair is called the sex chromosomes. So just as one last note, I think it's important that you spend some time learning the difference between sex cells and sex chromosomes, because oftentimes students confuse these, so it might be a good idea to actually compare these, and if you don't know the differences or similarities, then what you need to do is look it up. Okay? So I hope that this has helped, and at least you have some questions that you can search answers for or ask your friends to be better prepared for this test. I think that's it.